Thank you very much, Dan May, and we're really sorry for the technical problems with your talk. Um, Now we will open this up to different questions from the Slido, and we will be able to see all the members of the panel. Unfortunately, we've been having some problems seeing a couple of the speakers, including Jean-Laurent. So he will be able to take questions by audio. So I'm going to start from the questions that we have, and I'll start with Anna. A question from Slido here is, first of all, fascinating talk. Is MUC1 overexpression in COVID-19 infected lung a factor in the formation of hyaline membranes in damaged lungs? And maybe just to append to that question, you could tell us a little bit about the clinical trial you're doing.
Thanks. I'm not doing the clinical trial. This is a trial that has been initiated in response to our work and the work of another group from the Netherlands that independently found fostamatinib to have an effect in dampening down the hyperinflammatory response in COVID-19 in vitro. And so there was some evidence that was suggesting that fostamatinib may be of benefit, and our colleagues at Imperial College are doing a trial. So, and it's an investigator-initiated trial. I have no relationship to Rigel, who makes this drug, but I'm just gratified that our work may in some way help patients down the road, and that will be tremendous if something is found out of this. It's an ongoing study at the moment, so there are no results. Now back to the lung injury, there is an entire history of the role of MUC1, otherwise known as KL6, in not only correlating with the severity of ARDS in patients with severe acute lung injury, but also being predictive of the severity of ARDS in patients. And this is not just in response to viral infections, but other causes of ARDS. And so it has been long known that MUC1 has a role in acute lung injury. And it's not just a biomarker, but potentially causally related to the excessive amount of mucus that prevents the ability of the lung epithelium to repair itself and provide gas exchange. So we are hoping that these findings, which suggest that fostamatinib removes MUC1 from the plasma membrane of epithelial cells, will be of benefit.
Can I just, a brief follow-up question? There's been some recent papers published in the bioarchive around mucins and how they could be beneficial. Other, maybe not MUC1, but other mucins could be beneficial to protection from different viruses through interaction with glycoproteins. What are your thoughts on that?
Yeah, I think that as is often the case, and again, this is definitely true in the kidney where I have much more expertise, but too much of a good thing can be a bad thing. And I think that in the case of, just as in the case of, mounting an immune response can be good in taking care of the virus, but mounting a tremendously prominent response and severe inflammation obviously can be damaging. So I think it's very much in the same way. Mucins are meant to create a barrier that, especially both in the lung and in the kidney, in the epithelia, in the gut as well, protect from infection. But in the case of an excessive amount of MUC1, as has been shown many times over in acute lung injury, it is felt to be counterproductive and contributing to the pathogenesis. And that would be true for mucin 5A, which is also highly expressed in the lung as well.
So the drug that you're repurposing has high specificity for mucin 1, or does it target multiple mucins, or do you know?
I do not know the answer to that. We haven't tested that.
Great. We have another question from Slido here for Dan Meng. And the question is related to SARS-CoV-2 causing heart injury, which might be relevant to the complement system overreaction. Do you think that could be a possibility? In other words, SARS-CoV-2 generates such a high degree of heart injury that the complement system, platelets, and other parts of the complement system overreact. Denmeng, I'm sorry, you're on mute. I think we have to unmute Denmeng.
Yes, sorry. You mean the heart injury caused by SARS-CoV-2?
CoV-2.
Yes. The next. Sorry, I could you please?
So we posted the question in the chat for you to have a look at.
Okay. Okay. Oh, okay. Okay. Might be since the analysis, the single cell RNA-seq, there is indicated C1R expression is highly expressed. So I think the complement system is overreacted, but it should be further confirmed.
Understood. Okay. I have a question for Henrik. Henrik, we saw that at least you suggested that HIV infection, if I understood correctly, is anti-correlated with SARS-CoV-2 infection in the gut. Am I getting that right? And we see. Sorry, I think you're on mute, Henrik.
Right. Yeah. So in the HIV infected individuals, whereby we did the analysis for entry receptors, they were actually fewer cells that express those receptors, and also particularly downregulation of ACE2. So we haven't actually been able to study that in a true co-infection patients where you both have SARS-CoV-2 infection in combination with HIV. That's studies we want to do to see whether we actually see any exacerbation with the two viruses present at the same time. But we know from sort of bigger studies coming out of Cape Town that the big question was, does HIV infected people is that really a vulnerable population in COVID-19? And I think the data shows roughly like an odds ratio of 1.8 compared to diabetes, which sits between 8 and 10, I think, in the same population. So it's actually surprisingly not an extreme sort of risk factor.
Do you think that because there's also published work that ACE2's isoform in the gut is quite different from the one that's expressed, say, in other cell types like the lung, and that may play a role in the difference in the co-infection that's you're measuring in the lung? Yeah.
Well, that's a really likely situation, I guess, if you have different isoforms of ACE2 there, that could also sort of change the susceptibility to infection. And I know from colleagues that and also other studies, when you look at actually viral shedding from the gut, it's they're really not that infectious. I mean, there's been a little bit of contradiction, but a few studies have shown infectious virus. But majority really you don't see very much infection virus coming out of the gut, and that could probably be due to the environment, I guess, that the virus is simply just too fragile. So I think the more link could really be to sort of the general immune sort of link between the gut and lung axis potentially, where you really see an impact maybe on your type 1 interferon response, as suggested in other reviews.
Danming, the data you have for viral infection kind of is contrasting to what Henrik showed. You showed that actually MERS and RSV kind of enhance the infection. So what do you ascribe that to, the kind of divergence between HIV and MERS and RSV?
Yes, it's very interesting. In my opinion, maybe some patients with HIV have a drug with antivirus. So I think maybe after the drug treatment, the ACE2 expression is downregulated. I'm not sure whether HIV infection patient with the treatment or other treatment will have any effect on the ACE2 expression. I think it's a very important issue to determine how the SARS-CoV affect the HIV or other virus just mentioned the co-infection of HIV and SARS-CoV. What's the consequence? It's a very interesting work to determine. I think so.
Well, maybe I can comment.
Sure.
Maybe the difference. I mean, we could look at the antiretroviral naive patients, or patients who don't have received HIV treatment. And in just in those microscopy data, we didn't see any major difference. It could also be that HIV infection drives a different differentiation states of your epithelial subsets than you would see in these other respiratory infections. So I think that different scenarios that could explain the difference in expression.
Okay, maybe the different viruses have different effect on the ACE2 expression. Thank you.
Okay, we have a few more minutes left. There were a couple questions here for Jean-Laurent, but unfortunately he's left the panel. So maybe some of you may want to contribute to some answers on this. And I think it's about this convergence of the interferon pathway. Would any of you like to speculate whether GWAS data would find other common variants in the interferon-1 pathway that would be linked to the disease severity? I think, Henrik, you tend to suggest that because of prior HIV infection, that could raise interferon levels and be protective for subsequent SARS-CoV-2 infections. How plausible do you think that is? And maybe similar GWAS variants that are protective of people in HIV would somehow be protective for people in SARS-CoV-2? At least the interferon linked one.
Yeah. I mean, I think it's just overall sort of morbidity in HIV infected cohorts. It's also important to control for studies like population level studies that we done in terms of other comorbidities like diabetes. That actually it's actually you have less diabetes in the HIV infected population, but that could be also different levels of clinical care, which is a completely different setting. Type 1 interferon response. I mean, maybe the immune system is already set up in HIV infection for a more rapid type 1 interferon response that potentially could maybe change the kinetic of the response to SARS-CoV-2, but it's purely speculative. Yeah.
Anyone else want to contribute an answer to this question? Dan-Meng?
No, sorry.
You don't have to. You don't have to. Okay. There's the last question that we'll ask now, which is, again, from the Slido. Is it possible to track virus evolution in specific cell types? Considering, most of the cell types that have been covered in the session are well outside the lung. In terms of a natural history of virus evolution in the cell type, is that something that is plausible in kidney, in the gut, in the heart, and in other cell types?
Well, I don't know. I mean, I don't think I can comment too much. It's probably more like a technical question whether you can actually you can get enough viral sequence data from single cell studies in actually infected tissue, and thereby comparing different viruses and different cell types. I don't know whether that different cell types would drive different viral potential escape potentially. But I guess that's probably more like a technical question for the RNA-seq community, maybe.
Okay, I think we'll end our session here. Thank you very much. We're gonna have, I think, a recap of the day from Sarah Teichmann. Thank you very much for joining us for this session.
Thank you.
Thank you, Musa, and thank you all the speakers of this session. It was a fantastic second session and really super exciting. And thank you everybody for attending. I know that this has been incredibly popular, and there have been hundreds of participants watching online. So thank you to everybody. Next slide. Yes, and also to the chairs, John and Musa, special thanks. And also for the program committee. So it was myself and with Aviv, Diane Taylor, Bruce Arno, and Gian Marioni who put this program together. And thank you to my colleagues who may be online. And thank you to Brian, the video production team, and also the HCA executive offices. So John Randall, Tracy, Jenny, Anya, and Christine for the logistical help. So a heads up for the second day of the symposium, and that's Friday, 9th of October, so next Friday. And please register online to join. And these are the resources again at humancellatlas.org that I mentioned earlier. And next slide. Yeah, there'll be recordings both on YouTube and Bilibili. So all the talks are recorded for people who missed any session or want to go back. So this will all be available online. And let us know your ideas and your feedback. If you go to the polls at slido.com, you can feedback what you liked and what you didn't. And next slide. And that's it. Thank you, everybody. Have a great day or good evening, and see you next Friday. Bye.